Hello everyone and welcome back. So today let us look at enzymes in general and what are coenzymes and isoenzymes. Okay. So before we begin, let us first try to understand what are enzymes. Okay, so enzymes, as you already know, that they are biocatalysts which increase the rate of the reaction or velocity of chemical reaction without itself undergoing any change in the overall process. Okay, so enzymes are commonly known as biocatalysts and they increase the rate of the reaction without themselves undergoing any change. Okay, so what are the characteristics of the enzyme? Now almost all enzymes are proteins. Okay, so we all know that proteins are the functional unit of our body and enzymes also are made up of proteins. They are heat labile. Just like proteins are heat labile, they are also heat labile. That means temperature affects the working of an enzyme. If you increase the temperature, the enzymes will undergo denaturation because they are proteins. Okay, proteins and also undergo denaturation. So the enzymes, they will not work. So they are also heat labile. That means they work under optimum temperatures. Okay. Next, they are water soluble. So all the metabolism in our body takes place under water soluble conditions only. Okay, we require the reactions to be water soluble. So enzymes also are water soluble. Now they can be precipitated by protein precipitating agents. Okay, so all agents which bring about protein precipitation, that will also affect enzymes. Why? Because enzymes are made up of proteins. Okay, so proteins are precipitated by which agents? They can be precipitated by physical agents like heat, vigorous shaking, or chemical agents. Okay, so that we will study later. So what you should know is that all the precipitating agents, which protein precipitating agents, will also affect enzymes. And because they are made up of proteins which contain nitrogen, so they roughly weigh about 16% weight as nitrogen. Okay. Now let us look at classification of enzymes. Now the classification of enzyme is given by International Union of Biochemistry and Molecular Biology, and enzymes are classified into the following classification. The sequence of classification again is very important. So the first one is oxidoreductase. Okay, so as the name suggests that these undergo oxidation reduction reactions. So the enzyme, what it does is it transfers either hydrogen, okay, or addition of oxygen will take place. So oxidoreductase enzymes, they either transfer hydrogen or they add oxygen. The examples are lactate dehydrogenase, okay, which transfers hydrogen, or dioxygenase, which helps in by addition of water. The next is transferases. Now these enzymes, they transfer groups which are other than hydrogen. Okay, hydrogen is already taken care by oxidoreductase. So the other groups apart from hydrogen, they are transferred by the enzyme transferases. Okay, so transferases, the transfer group other than hydrogen. Okay, for example, aminotransferases, they transfer the amino group, okay, NH2 group. Next we have is hydrolases. So as the name suggests, hydrolases are enzymes which breaks bonds with the help of water. Okay, so hydrolases, they bring about cleavage or breaking of bonds with the help of water. Example is all the digestive enzymes in our GI tract. Okay, most commonly trypsin, and acetylcholinesterase is another example. Next is again cleavage of bond but without the addition of water. So these are lyases. Okay, lyases also bring about breakage of bond but without the addition of water. For example is aldolase and HMG coenzyme A lyase. These are enzymes which are lyases. Next we have isomerases. Okay, so these enzymes, they produce optical, geometrical or positional isomers. Okay, so these bring about change in isomers or isomerization. Example is racemases and epimerases. Next is ligases. Now ligases, they bring about condensation of two groups. okay, which are ATP dependent. So ATP dependent condensation of two molecules, okay, binding together, not breakage. This involves binding together. So condensation of two molecules. The example is acetyl coenzyme A carboxylase PRPP synthetase. Okay, here because the two groups are joined together, okay, so here ATP also is utilized. And the last one is translocases. Okay, so these enzymes help in transfer of groups across the cell membrane. Okay, example is ATP ADP translocase. Okay, so these are the classification of enzyme, okay, based on the function. Now let us look at what are coenzymes. Okay. So coenzyme, as name suggests, these are a non-protein part of the enzyme. Okay, so up till now we studied that the enzymes are proteins. Okay, so but enzymes are also made up of a non-protein part, which is called coenzyme. So if you look at the enzyme, it is made up of two parts. One is apoenzyme and the next is coenzyme. The apoenzyme is the protein part, whereas the non-protein part is the coenzyme. Okay, so what does apoenzyme do? It is the protein part. It gives the three-dimensional shape. Okay, now we know that proteins have structural organization of proteins are there into primary, secondary, tertiary and quaternary structure. So the functional protein is a tertiary structure which gives the 3D shape. Okay, so this enzymes we all know are mainly proteins. Okay, so the function of protein is by the 3D structure. So apoenzyme, which is the protein part of the enzyme, it brings about the 3D structure for the function of enzyme. Okay, and it is heat labile. Whereas the coenzyme, it is the non-protein part. Okay, coenzyme is the non-protein part. It is also known as prosthetic group. Okay, now these coenzymes, they help in transfer. Okay, now we have group 1 coenzyme and group 2 coenzyme. Group 1 coenzyme brings about oxidation reduction reactions and transfer groups like hydrogen. Okay, so these are the groups which help in transport of hydrogen. Okay, for example, NAD, NADP, FAD, etc. Okay, so if you see over here, lactate is dehydrogenated to pyruvate. Okay, so the hydrogen is removed. So this hydrogen is carried by NAD plus to form as NADH plus H plus. Okay, so the hydrogen is removed from lactate. It is taken up by coenzyme NAD plus. Okay, so and the enzyme which brings about this is lactate dehydrogenase. So lactate dehydrogenase is the enzyme which contains the coenzyme which is NAD, and this NAD is transporting the hydrogen or transferring the hydrogen group. Okay, so this is example of group 1 coenzyme. Okay, now group 2 coenzyme are enzymes which transport groups other than hydrogen. Okay, so the other coenzymes are, you can have thymine, pyrophosphate or active form of vitamin B1, TPP. Okay, then you have active form of vitamin B6, pyridoxal phosphate, biotin, coenzyme A, tetrahydrofolate, ATP. So all these are coenzymes and they transfer these specific groups. Okay, for example, pyridoxal phosphate is the coenzyme which transports the amino group, or biotin transports the carbon dioxide group. Okay, so groups other than hydrogen are transported by group 2 coenzymes. Okay, now this coenzyme is binding loosely with the protein part. Okay, so the bond between the protein part, apoenzyme, and coenzyme is a loose bond. Okay, and it can be broken. So coenzyme can break from this apoenzyme and form bond with another apoenzyme. Okay, so most see over here, so that these coenzymes can be reutilized. Okay, so the bond between apoenzyme and coenzyme is weak. Okay, this can be broken and these coenzymes can be reutilized with other apoenzymes. Now most of the coenzymes are made up of vitamin B complex. If you see over here, these coenzymes are mainly B complex vitamins. Okay, so how do they function? That we will study in vitamins. Okay, so this is excuse me, these are the functions of apoenzyme and coenzymes. Now, when apoenzyme and coenzyme they combine together, it is known as holoenzyme or a complete enzyme. Okay, so holoenzyme is a term which basically includes apoenzyme and coenzyme or a complete enzyme. Okay. Now we have another enzymes type of enzymes which are called metalloenzymes. So metalloenzymes are nothing but enzymes which require metal ions for their activity. Okay, so those are called metalloenzymes. Okay, enzymes plus metal ions. Now which metal ions? So these are the common metal ions. Zinc is a metal which is required by the enzyme. Carbonic anhydrase, alcohol dehydrogenase for its action. Similarly, magnesium, manganese are required by enzymes which are involved in carbohydrate metabolisms like hexokinase, phosphoctykinase. Okay, and copper is present in or required for the enzyme tyrosinase, cytochrome oxidase. Iron is required for cytochrome oxidase, catalase, peroxidase. Okay, so these are the metals which are required for functioning of special enzymes. Okay, so these are called metalloenzymes. Now we have another term which is cofactor. Okay, so cofactor is a broad term which includes all coenzymes as well as metal enzymes. Okay, or the non-protein part. Now let us look at isoenzymes. What are isoenzymes? Now isoenzymes are different molecular forms of the same enzyme. Okay. Now if you look at the LDH enzyme, okay, so the LDH enzyme is made up of subunit A and subunit B. Okay, so the structural component of LDH1 and LDH5 is different, but they have the same function. Okay, so different molecular forms of the same enzyme or enzymes which have different structure but same function. Okay, if you look at this LDH5, this is made up of subunit A, which is different from LDH1, which is made up of subunit B. Okay, but both LDH1 as well as LDH5 have the same function. Okay, so these are LDH1 and LDH5 are isoenzymes. Similarly, we have is LDH2, 3 and 4 also. Okay, so different structural forms of the same enzyme are called isoenzymes. Okay. Now all different molecular forms of isoenzymes may be seen in the population. Okay, so for example LDH1 to LDH5 all are present in all of us. Okay, or we can also have various proportions of isoenzymes present in different population. Okay, so that we will see. Now different isoenzymes are located in various tissues and an increase in the subtype indicates a tissue abnormality. Okay, for example again let us see LDH1. Now LDH1 is made up of subunit B. Okay, and LDH5 is made up of subunit A. Now LDH5 is produced in some other organ, let's say organ A, and LDH1 is produced in organ B. Okay, so if there is excess of LDH1, so this indicates that the organ B is where it is produced. Excess is produced from this organ B, or a dysfunction of this particular organ B has occurred. Okay, similarly if we have excess of LDH5, okay, so we can assume that since LDH5 is produced in organ A, so there occurs a dysfunction in organ A. Okay, so this is the clinical enzymology or importance of enzyme. Okay. So this is by measuring the enzyme activity we can know which isoenzyme is more active or which isoenzyme is present in more concentration, and that indicates that that particular organ is dysfunctional. Okay. Now isoenzymes can be separated by electrophoresis. Now to understand that whether LDH1 is increased more or LDH5 is increased more, we need to separate the LDH. Okay, so in blood when we measure LDH, we measure the total LDH. Now that total LDH is made up of LDH1, 2, 3, 4, 5. Okay, so it is a combination of all these isoenzymes and we get one single value in the blood. Okay, for example, if the LDH value is 500, so it is made up of total of LDH 1, 2, 3, 4 and 5. Okay. So, and if the LDH value is elevated, it only tells us that LDH is elevated. It does not tell us which of these subfractions or which of the isoenzymes of LDH is elevated. Okay, to find out which of the isoenzyme is elevated, we need to separate these isoenzymes. So we need to separate the LDH into its constituent isoenzymes. So that is done by electrophoresis. It's a separation technique which separates charged particles. Okay. So once we do the electrophoresis, we can separate the isoenzymes. Okay, so LDH 1, 2, 3, 4, 5 is separated. Once they are separated, we can find out whether this particular isoenzyme or which isoenzyme is more than normal in concentration, and whichever isoenzyme is more than normal in concentration, it indicates the dysfunction of that particular isoenzyme and where it is located. Okay, so this is how we can utilize the knowledge of isoenzymes and trace back to its origin and find out the organ dysfunction. Okay, so more we will study about this when we talk about diagnostic importance and clinical importance of enzymes. Now, isoenzymes can be heat stable. Okay, isoenzymes, some, as we all know that proteins are heat labile. Okay, so some of the isoenzymes, okay, can also be heat stable. Okay, for example, bone alkaline phosphatase, because alkaline phosphatase is present also in the liver. It's also present in the biliary tract. Okay, but they are heat labile. They are affected by heat, whereas the bone alkaline phosphatase is heat stable. Okay, so using this property of heat, we can isolate or separate bone alkaline phosphatase from other isoenzymes of alkaline phosphatase from the biliary tract or the liver. Okay, similarly, okay, we also have tartarate labile acid phosphatase, another isoenzyme, okay, which is acid phosphatase, which is inhibited by tartarate. Okay, so we can use this tartarate to inhibit a specific isoenzyme of acid phosphatase. Okay. Next, we have isoenzymes can also have different Km value or affinity for the substrates. Okay, for example, glucokinase and hexokinase. Now, both of them act on glucose. Okay, but the affinity or the substrate affinity of glucose for these two isoenzymes is different. Okay, so hexokinase is having more affinity, glucokinase is having less affinity. They also differ in their cofactor requirement. Okay, so these are isoenzymes also based on their individual property. They can be separated or they can be identified. Now we have isoenzymes. It can be either true isoenzymes or it can be an alloenzyme. Okay, so true isoenzymes are enzymes in which they are produced from different genes. Okay, for example, the amylase enzyme. Now, amylase can be produced. We have two types: salivary amylase and pancreatic amylase. Okay. Now, because it is an amylase, it has both of them have same function. Okay, but these two isoenzymes are produced from different genes. Salivary amylase is produced from different gene. Pancreatic amylase is produced from different genes. Okay, so they are called true isoenzymes, and both these amylase have the same function, that is hydrolysis or breakdown of starch. Okay. Whereas alloenzymes are enzymes which are produced from the same gene, not different gene. Okay, for example is glucose 6-phosphate dehydrogenase. Now this is an enzyme which is present in the HMP shunt, okay, which is one of the metabolic pathways of carbohydrates. So glucose 6-phosphate dehydrogenase is an example of alloenzyme. Now the gene which produces the enzyme glucose 6-phosphate dehydrogenase is an X-linked gene. It is present on the X chromosome. Now variation in that gene which is present on the X chromosome can bring about 400 different types of G6PD. Okay, why? Because the gene produces different types of G6PD based on various types of mutation and other causes. So the same gene is producing different types of isoenzymes of glucose 6-phosphate dehydrogenase. Here isoenzymes are produced from two separate genes. Okay, so we have two isoenzymes which are produced from two separate genes. Here we have around 400 types of isoenzymes but they are all produced from same gene. So this is the difference. Okay, so these are what isoenzymes are and what are its importance. Okay, so thank you very much. I hope you have understood, and I will see you soon with the next lecture. Okay, so please like, subscribe, and share my work. Thank you.